This is the ls230 with variable speed module. This is predominantly used for running samples in water. Generally you would enter the sample into the sample vessel there. Everything else is done via the keyboard and the software which runs on the PC. So it's all under software control. If we go up to the screen there, if we want to run, we simply click on run on the screen. Hopefully you can see. Got a drop down menu and call up run cycle. You ask it, we would advise you to ask it to measure the background, to measure the loading, which is entering in the sample information, and then the run settings, and then simply press start. First of all, you get a background screen up, so this is basically looking at the fluid which is passing through the optical cell and eliminating any of the particles that may be in the background. It takes a little while just to set itself down while it's waiting. The status bar towards the top of the screen shows you what is actually going on, so it's measuring background at the moment. So each of the 126 detectors has its background measured, and then that background level of light diffraction is subtracted from the sample, so that you just have the light diffracted by the sample itself. Once the background is finished, we're ready to add the sample, and it will typically bleep at you to let you know that, and the measure loading screen comes up, which it does now. If we were now to be using the system, we would add the sample into here, and then once we've added sufficient to get the right level of what we call obscuration, then we click done on the screen, and we have a enter sample information window drop down where we can enter any information we want about the sample, how we want it stored, any comments like dispersed or liquid that it's running, and then click OK. If we want to, we can look at the run settings, how long we run for, how many runs we make, whether we want to do one or do several runs and average them. Once we've decided all of that, we click OK, and the instrument carries on making the measurement. Size distribution is displayed automatically. In this case we've got one that's quite small on the left hand side here, differential and cumulative information, which will continually update during the sample analysis. Once the 1 minute of sample analysis in this particular case is finished, we'll be able to do further analysis such as statistical manipulation on the data, get means, modes, medians, that type of thing, D10, D90, or we'll be able to change the display, overlay 2 or more sets of data so we can see if samples are different one to the other. But basically once the sample is stored, you can recall it any time, play around with it, play with the graph, do what you like in terms of data manipulation. And that's really all there is to it. Everything is done from the keyboard apart from the actual introduction of the sample into the sample vessel. And that's it. Okay.